So it's a great pleasure for me to be able to introduce the next speaker, who is the winner of this year's Beddington Medal for the BSDB. So the Beddington Medal is awarded to the best PhD thesis from the previous year. So the Beddington winners are the future Waddington winners. And in this case, it seems particularly apposite to compare the two, because the winner this year is John Robert Davis, who has completed his PhD in Brian Schramer's lab at King's. And what really impressed us in his application for the medal is the just beautiful combination of fantastic live cell imaging with theory, with computational modeling, to address a really exciting big question in developmental biology. And I think this is exactly the kind of thing that would very much appeal to Lewis. So I think, as I say, that there's every possibility that we'll all be back here in 20, 30 years' time awarding the Waddington Medal. But right now, it gives me great pleasure to welcome John Robert onto the stage to receive his medal and to present his talk, which is entitled Intercellular Forces Orchestrate Cell Repulsion in Embryonic Pattern Formation. Thank you.
As I'm loading this so that there's not some awkward silence, let me introduce myself. I am John Robert Davis, and I've been doing a PhD in Brian Schramer's lab. First of all, I'd just like to thank the committee for awarding me this year's Beddington Medal and inviting me to speak in front of all of you here today. It really is a huge privilege. I'd also like to thank the BSDB, the BSCB, and the Company of Biologists for all the support that they've given me throughout my PhD, mainly in helping me go to conferences and workshops. Ottoline's introduction was very kind, but I would say that a lot of this work is down to my PhD supervisor, Brian Schramer, who at times dragged me through this PhD, and I'd like to publicly thank him for all the support and encouragement he's given me over the last few years. It's been a huge pleasure working with him. He's sitting by the end of the aisle. If you ever know Brian, he always sits at the end of the aisle because he has a bladder the size of a walnut. So I'm going to talk to you today about the work that I did in Brian's lab on this relatively obscure phenomenon known as contact inhibition of locomotion. So contact inhibition was discovered by this man, Michael Abercrombie, over 60 years ago. And we're very fortunate in the Schramer lab to have some of the original movies he used to characterize this phenomenon. So Abercrombie pioneered time-lapse microscopy, and he was interested in chick heart fibroblasts migrating in vitro. And what he noticed was that when a migrating fibroblast came into contact with another cell, it would stop its forward motion, repolarize, and then migrate away in the opposite direction. And he termed this contact inhibition. And in the years following, many other cell types have been said to undergo this process. However, we still don't know how it's regulated and if the mechanisms of regulation are the same in different cell types. So why were we interested? Well, when Brian was a postdoc in Paul Martin's lab, he was working with Drosophila embryonic hemocytes. So here I've got a bright field image of the Drosophila embryo, and we're looking at it along the ventral surface. And I've labeled the hemocytes with this red nuclear marker. Now, hemocytes are professional phagocytes, so they have an important role in the immunity of the fly, but they're also required for development, where they clear away apoptotic debris and lay out extracellular matrix throughout the entire embryo. However, they're derived in the head mesoderm early on in embryogenesis, and therefore the rest of development they're spent migrating throughout the whole embryo, and they do this via specific migratory routes, and one of which is along this ventral surface, where initially they move along this midline, and once that's become fully populated, they then disperse laterally and acquire a three-line pattern. Now, it's important to note that once these cells have acquired this three-line pattern, they don't stop moving. But these cells are highly migratory, and they're constantly moving and bumping into each other and undergoing this process of contact inhibition. Now, all of the work that I'm going to show you today is of hemocytes migrating along this ventral surface within the Drosophila embryo. So, when Brian looked at these collisions in a bit more detail, he realized that the microtubule cytoskeleton was playing an important role in hemocytes' ability to repel one another. And what he noticed was that during collisions, the microtubules of cells that were undergoing a collision would come together and form bundles, and these bundles would align between these two cells. And this alignment of these microtubule bundles seemed to initiate this repolarization and migration away. And if he overexpressed a microtubule severing protein such as spastin in these hemocytes, what he found was that the cells were unable to repel each other and they stayed in contact for longer. But what he also noticed was that if he disrupted this microtubule bundle alignment, then the hemocytes failed to form this three-line pattern, which made us think, well, possibly contact inhibition was playing a role in the involvement of the hemocyte's ability to acquire this three-line pattern. However, we were not too sure about the exact nature of this role. Like, was contact inhibition a mechanism that drove the emergence of these three lines? Or was it a mechanism that allowed the hemocytes to maintain the pattern once it had already been laid out by extrinsic cues? And when I started my PhD in Brian's lab, that was the question we very much started to try and address. How critical is contact inhibition in hemocytes' ability to form these three lines? Now, when I started, Brian had already shown this work to a retired professor at King's College London, Graham Dunn. Now, Graham Dunn not only did his PhD under Michael Abercrombie, so was well versed in the field of contact inhibition, but spent his whole career analyzing and modeling cell migration. And he's one of my heroes in science, and he came up with what I consider one of the most elegant models I've ever seen in biology. And he said to us, well, in order to assess how critical contact inhibition is for this dispersal process, why don't we come up with a simple mathematical simulation of hemocyte dispersal based purely on the migration dynamics of freely moving and colliding hemocytes? Now, I'm not going to go into too much detail about this work because it's been published, but essentially what we did was by using the nucleus as a fiduciary marker, we were able to automatically track the migration of these cells. And by looking for the observation of whether these microtubule bundles aligned, we could tell if a cell was freely moving or colliding. And through that, we came up with these two mathematical equations to explain the motion of freely moving and colliding hemocytes. Now, I want to state here that all of the parameters that went into these equations are derived from this automatically tracked data. And the only other parameter that we have in this model is this internuclear distance of 30 microns, which we use as a switch to move from one state to the next. So when cells are further away than 30 microns, then they're freely moving. But if they come within 30 microns of another cell, they start to then behave as if they're colliding. And the reason we can use that 30 micron distance is what I haven't told you, is that when these microtubule bundles first align, there is always invariably 30 microns between the nuclei of these colliding cells. Now, we don't know exactly how this process is governed, but it's very consistent from collision to collision, suggesting that it's tightly controlled. So when we simulate hemocyte dispersal in an embryo of comparable size, shape, and cell number to reality using our model, we found we were able to mimic the timing and the formation of this dispersal pattern, which suggested that contact inhibition of locomotion was driving the emergence of this three-line pattern. And what was happening was that the cells were trying to optimize the space between themselves and their neighbors, but due to the confines of the Drosophila embryo and the amount of hemocytes present within this space, it forced them to acquire a three-line pattern. So after we published this work, we then started playing with several of these parameters in the model to see how robust this final pattern is to those parameters. And one of the parameters we looked at is this here, which is psi-RT. Now, this just simply states how repellent the cells are from each other. So a high value means that the cells are highly repellent and they want to get as far away from their colliding partner as quickly as possible. And a low value means that the cells, well, they're not that bothered about their colliding partner, they can persist in contact for a lot longer, and they're more likely to just skirt past each other. Now, from real data, we found out that this value was very high in hemocyte collisions, suggesting that these cells were highly repellent from each other. What we also found was that it was very consistent from collision to collision, suggesting that this process was also tightly controlled during these collisions. So we wanted to know, well, what happens if we randomize this repulsion dynamic? So cells could arbitrarily choose how repelled they were by their colliding partner. So some cells could be highly repellent and other cells just not bothered at all. And when we did this, what we found was that the hemocytes in this random simulation failed to properly disperse, let alone acquire any kind of pattern whatsoever, suggesting that in order for contact inhibition to drive the immersion of this embryonic pattern, collisions needed to be, or the repulsion dynamics needed to be tightly controlled in these cells. So we wanted to go to real collisions and see if this was the case. And we analysed this in lots of different ways, but the simplest and my favourite was just to measure the distance between the nuclei of colliding hemocytes. And we wanted to extend this analysis over time, but we knew that collisions were of variable time spans. So we used as a temporal register this time point of microtubule alignment, which we set as our 0 time point. And when we did this, what we found was that collisions were actually a series of changes in the migration dynamics of these colliding hemocytes. So initially, the cells are slowly moving towards each other, and now this is a bit of an artifact by the fact that this analysis is only done on colliding hemocytes. However, 2 minutes before these microtubule bundles align, we see that these cells all of a sudden speed up towards each other. Upon microtubule alignment, they then slow down, and this is followed 2 minutes later by the cells repolarizing and then migrating away from each other. Now, this suggested to us that hemocyte collisions were actually a choreographed cellular dance, a bit like the first dance of a bride and groom on their wedding day that they've practiced for months. And what they are not is the kind of haphazard drunken dancing that you see later on in the evening when people have had a few drinks. Now, if anybody saw me dance last night, you would have seen me do very similar moves to this. That's because this is my sister, and we both learnt dance from the Lord of Dance, or as I like to call him, Dad. So we wanted to know what could be initiating this choreographed cellular dance. And the first thing we looked at was the actin cytoskeleton, because we knew from previous work that the lamellae of these colliding cells came into contact before these microtubule bundles aligned. And when we measured the time between lamellae contact and microtubule alignment, what we realized was that it precisely coincided with this time that these cells all of a sudden moved towards each other. Not only that, but when we looked at the time between microtubule alignment and lamellae separation, we saw that this preceded any repolarization and migration away. And it suggested to us that the actin cytoskeleton was somehow initiating and possibly orchestrating this choreographed cellular dance. So we wanted to look at these actin dynamics in a lot more detail during collisions. So here we have a time-lapse movie of a hemocyte freely moving along the ventral surface, and we've labeled the actin cytoskeleton with LifeAct GFP. And hopefully what you can appreciate is that this is a very highly dynamic structure. So the actin network is assembled at the periphery of the cell, and it's disassembled at the rear near the cell body. And this assembly and disassembly drives the motion of the actin network from the periphery towards the cell body, which is known as retrograde flow. And this retrograde flow is also aided by myosin 2, which pulls on the network, contracting it towards the cell body. Now, it's this retrograde flow which is thought to be the motor that drives cell motility. So we wanted to quantify these dynamics. And this is when another hero of mine in science, Andrei Lukic, who is another PhD student in the Stromer lab, came along. Now, he has an engineering background, and he developed a technique which he likes to call FLUPS, which is fluorescent pseudospeckle analysis. And it's very similar to the kind of PIV algorithms that other people have used to study actin dynamics in vitro. And what this FLUPS analysis allows us to do is to quantify these dynamics and represent it as a heat map, where the color represents the rate that this network is moving, and we can overlay arrows which point in the direction that that network moves in. So we wanted to extend this FLUPS analysis to colliding hemocytes. So first of all, we just looked at time-lapse movies where we labeled the actin cytoskeleton just to have a look to see what was going on. And what we found was that there was a dramatic and dynamic reorganization of the actin networks as these cells were in contact. And that this reorganization seemed to focus around an actin fiber which forms between the lamellae of these two cells, like across both lamellae. And as these cells separate, what we find is this actin fiber is buckled and then brought back to the cell body where it is disassembled. So we wanted to quantify these dynamics. So we ran those collisions through Andrei's FLUPS analysis, and what we found was that as these cells were in contact, a corridor of low retrograde flow developed between these two cells. Now, this corridor of low retrograde flow co-localized with the position where this actin fiber was forming. And I'm not going to show you the evidence today, but it's also the region where the microtubules are lined between these two cells. Now, as these two cells separate, this corridor of low retrograde flow disappears, and the retrograde flow rate increases. And it increases to values that you very rarely see in freely moving cells. So not only did we see this development of this corridor of low retrograde flow, but we also noticed that the direction that the network flowed also changed during collisions. So when cells are freely moving and prior to collision, the actin network moves from the periphery towards the cell body. And you can see that by these arrows here. However, as this corridor of low retrograde flow develops, we see that the direction of flow reorients, and that now flows from the periphery of the cell where it's still assembled, but it flows towards this region of low retrograde flow. So the actin network is now being moved towards this region, and this seems to coincide with the formation of this actin fibre. So we wanted to know, well, what could be initiating this global reorganization of this actin networks? And the first thing that we looked at was to see whether an adhesion formed between these two cells as they came into contact. And we used as a marker for adhesion this protein zixin, which is a known component of cell-cell adhesions as well as cell matrix adhesions. And what we found was that as the lamellae of these cells came into contact, a puncture of zixin developed between the two cells, and this persisted for the entire time that the cells were in contact. And when we quantified the actin dynamics with respect to this zixin puncture, which we pseudo-colored in white here, we see that this reduction in the retrograde flow occurs after this puncture has formed, and this corridor of low retrograde flow develops in line with this adhesion. And what we think is going on here is a mechanism that's analogous to the integrin clutch idea thought of in cell migration, by which an adhesion is engaged in the actin cytoskeleton and then causing a reduction in its flow. Now, Andre, when he saw this, wanted to ask the question, well, is this adhesion physically coupling these two cells as they come together? And in order to analyze this, he did a correlation coefficient analysis. And what this basically does is he looks at changes that are happening in the actin network in one cell and sees if they are also occurring in the other cell simultaneously. And when he did this, what we found was that prior to these two cells coming into contact, there's a very low baseline level of this correlation coefficient. However, upon lamellae contact, we see that this dramatically increases, suggesting that as this adhesion forms, these cells are becoming physically coupled. Now, this correlation diminishes somewhat over time, throughout the time course of the collision. However, at the time of lamellae separation, as we saw this spike in retrograde flow rates, we see a second peak in correlation between these two cells. So far what we see is that these cells come into contact and adhesion forms. It reorganizes the actin network, and it drives the formation of this actin fiber across both cells. So we wanted to look at the nature of this actin fiber in detail. And one thing we did was look at myosin II localization during colliding cells. So here we have two hemocytes undergoing a collision, and we've labeled the actin cytoskeleton, and also myosin, which you can see as these green punctate. Now what we noticed is that myosin II flows with the actin network. So as cells are undergoing a collision and the actin network flows towards this corridor of low retrograde flow, so does myosin. And as it reaches this region and coalesces along this actin fiber, we see that it forms this repeating punctate pattern. Now this is very reminiscent of stress fibers in vitro. So we wanted to look at other known stress fiber components to see if they were also localized along this fiber. And one that we looked at is diaphanous. Now diaphanous is a formin, and it's involved in nucleating actin filaments, and in vitro it's been shown to be required for the formation of stress fibers. And as this actin fiber develops, we see that diaphanous localizes along its entirety, which suggested to us that this is indeed a stress fiber-like structure. Now, as stress fiber-like structures are known to be contractile, it started to make us think, well, are these two cells pulling on each other as they come into contact? Now, coincidental evidence that we had that kind of supported this hypothesis was when we looked at the lamellar dynamics of these two cells. So here we have a chymograph of two cells colliding. So this is one cell here, and this is another cell here. And we've pseudo-colored regions of extension in blue and regions of retraction in red. And hopefully what you'll be able to see is as these two cells come into contact, they then persist in contact, but as they separate, there is a sudden and synchronous retraction between these two cells. And when we quantified the retraction rate in these cells, we found that it was far greater as these cells separated than it was in freely moving cells, and that this retraction rate diminished over time. Now, as the retraction rate is thought to be an indicator of the tension present within a cell, it suggested to us that possibly as these cells were in contact, they were increasing their tension. Now, in other words, what we thought was going on, that as these cells came into contact, they started to play a game of tug-of-war, but that this goes wrong, and the two cells snap apart. And I'd like to state here that this retraction seemed to occur before we see any new extension of lamellae. So we wanted to directly test this kind of tug-of-war gone wrong hypothesis. And the way that we did that was through laser abscission experiments. So what we wanted to do was we wanted to cut with a multiphoton laser this actin fibre. And we wanted to cut it in the region that these lamellae overlapped with one another so that we made sure that we definitely severed the link between these two cells. So therefore, when we wanted to compare this to freely moving cells, we decided to do two regimes. One in which we would make a small cut at the edge of the lamella, similar to what we were doing in these colliding cells, and another ablation where we would ablate actin fibers within the actin network. And when we did this, what we realized was that the retraction rate in colliding cells was far greater after we made the abscission compared to freely moving cells. And when we quantified this, we saw that it was twice that of freely moving cells, furthermore suggesting that there was an increase in lamellar tension. However, we work with a lot of theoreticians, mathematicians, and they're just not happy with this. And they wanted to infer the actual forces present within the lamellar networks of colliding cells and freely moving cells. And the way that we did that was, first of all, to assume that the actin network was a viscoelastic material. And this is a very common paradigm in labs that look at the actin network. And instead of measuring retraction rate, we decided to measure strain. Now, strain is just the percentage of the lamellae that's deformed from one frame to the next. And when we did that, we can come up with graphs like this. And what we noticed was that there seemed to be two phases to this retraction of the lamellae after we made the ablation. And we came up with this equation to explain this retraction. So initially, there's an exponential decay in this retraction rate as the two cells snap apart from each other. But what we see is that over time, the retrograde flow of this actin network kicks in and then just continues to retract the network towards the cell body. And when we fitted this model with our data, we saw that it fit really nicely. And what this now allows us to do is to extend our analysis and infer the strain present in the lamellae before we made the ablation. And from strain, you can then do a calculation to get force. And what we found was that the force in colliding cells was 3 times greater than in freely moving cells, suggesting that as these cells are in contact, they are indeed pulling on each other. But we wanted to know if the release of this tension was sufficient to induce this backward movement of the cells. So we performed more abscission experiments, where this time we also automatically tracked the nucleus to see where the cells would move to after we made the ablation. And we did this in freely moving and colliding hemocytes. But in colliding hemocytes, we also did 2 different types of experiments. One in which we did actually ablate the actin fiber between the 2 cells, and others where we pretended to, and to see where the nucleus would move if we just didn't do anything. Now, so we can perform these laser abscission experiments, we can see the recoil and then track the nucleus. And we can represent the movement of the nucleus as these velocity maps here. So these arrows just represent the direction and the distance that these cells, the cell body has moved 1 minute after we've made the ablation. And if we normalize this to the ablation site, which we represent as a red arrow here, we see freely moving cells, they just don't care, and they continue to move in a random direction. There's no preference in their direction after we've made the ablation. If we pretend to make the ablation, we see that the cells move towards each other. Now, this is consistent with the data that I showed you beforehand, that as these cells are in contact, they like to move as fast as possible towards each other. However, if we do make the ablation, what we found was that we were able to induce a rearward movement of the cell body, suggesting that release of tension is sufficient to cause these cells to pull apart. So we wanted to know, well, what happens in colliding hemocytes if we stop the ability of them to pull against each other? And we decided to do this genetically and look at collisions in hemocytes which were mutant for diaphanous. And what we found was that diaphanous mutant hemocytes failed to form this stress fiber-like structure. Not only that, but when we looked at their retraction dynamics, unlike control cells where we see this sudden and synchronous retraction, diaphanous mutant hemocytes don't show this. And often what you see is the cells separate and then go back into contact with each other. Furthermore, when we quantify these retraction rates in colliding and freely moving cells, whilst control cells we saw an increase in this retraction rate during collisions, this did not occur in diaphanous mutant hemocytes. So we wanted to know, right, well, what would happen to the repulsive dynamics of these cells if we've ablated the formation of this stress fiber? So here are some more velocity plots. So this is just the direction that the cells move 3 minutes after the microtubule bundles align. So this represents the time it's taken the cells to repolarize and have some time to kind of migrate away from each other. And in control cells, you see a preference to move away from the colliding partner, which we've represented here as this red arrow. However, in diaphanous mutant hemocytes, they fail to do that, and their migration is random, suggesting that they're no longer able to tightly control their repulsion dynamics. Now, if you remember, the reason why we started looking into collisions in so much detail was that mathematical model that suggested that in order for contact inhibition to drive the emergence of this embryonic pattern, the repulsion dynamics of hemocytes need to be tightly controlled. And here we have a nice genetic system in which we can test this hypothesis. So we looked at the patterning of hemocytes in embryos, control embryos, and those that are mutant for diaphanous. And whilst in control cells, the hemocytes space themselves out quite nicely and form this three-line pattern, we see in diaphanous mutant embryos that the hemocytes fail to do this. And you get regions where they coalesce together and other regions where you've got large spaces where the hemocytes aren't present at all. But not only can we look at a static image, but we can also look at the average position of hemocytes throughout this entire dispersal process. And when we do that, we come up with these, what we call domain maps. So each of these domains is the average position of a hemocyte throughout the entire dispersal process. And in controlled hemocytes, you can see that they nice and evenly space out between themselves. However, in diaphanous mutant embryos, this fails to happen whatsoever. And if domains do form, they're irregularly spaced from one another, suggesting that indeed, in order for this pattern to emerge, contact collision repulsion dynamics need to be tightly controlled. So just to sum up, so I've told you that collisions between hemocytes are a partially mechanically driven process. Now, this is not a new idea. Michael Abercrombie, in his original paper back in the 50s, came up with a similar hypothesis. And he said, as a result of the adhesion between them, they push or pull against each other to some extent. When adhesion breaks, the release of potential energy stored as elastic tension produces the absurd sudden acceleration. And here we have one of his movies of chick-heart fibroblasts, and you can see a similar snapping process. But this mechanical regulation of cell repulsion is what drives the emergence of this embryonic pattern. So it gives me enough time to just say thank you to everybody I worked with, especially Graham, Brian, who was a superb supervisor, and Andre. And I'd just like to leave you with a movie to show the kind of shenanigans that we get up to in the Stramer lab. So I present to you this movie, which I call the Stramer Twerk. And thank you very much for listening.
Thank you very much. Again, quick question.
Hi there, that was just fantastic. At some point, the hemocytes will find themselves in situations where now they don't want to repel each other, but they want to come together, like in infection or wound healing or metamorphosis. So at which point of the process do you think that the dynamics change?
So that's a great question. So we've done some preliminary experiments where we've wounded the epithelium along this ventral surface and looked at what the hemocytes do. And they do indeed switch off this contact inhibition dynamics. However, it's quite complicated. So the first cells that go to the wound seem to form an actin fibre, which is along the kind of like wound edge. And if other cells come in and try to get to that wound and the hemocytes have occupied it and they've got a bit of free lamellae that's not part of this kind of ring, then they're able to still somewhat contact inhibit one another. But there is something different about when they're actually at the wound edge. And that's one thing that Brian's looking at the moment, is to see what is kind of changing. Is it the actin dynamics? Is it the substrate they're working? Is there some kind of signal that's released that kind of diminishes this response?
Okay, thank you. I think everyone's probably dying for coffee. Shall we try and be back here by about 10 past 11, and then we'll start the next session at 10 past 11. And thank you very much for a really great talk.
Thank you.